Hello guys and welcome back to Excision Academy. So we were doing DNA repair and its mechanism. So till now we have discussed different types like direct reversal. We have discussed, right? In the direct reversal, we talked about the methylation, and we have also talked about the thymidine dimer, right? Or the pyrimidine dimer basically. Then we have discussed about the mismatch repair, correct? So in which we have talked about the mute SLH. And then later we have talked about the base excision repair, correct? Do you guys able to remember, recollect?
Yes, ma'am.
Okay. Now we'll be doing the another one, that is nucleotide excision repair mechanism. Okay. Like it just advice that you guys make a column of it, because कैसा होता है ना, जब पूरा पढ़ने के बाद at the end में confusion आ जाता है कि कौन सा प्रोटीन कौन से रिपेयर मैकेनिज्म का है, ठीक है? Like for example, in the base excision repair mechanism, we have talked about the glycosylases. An example of uracil glycosylase हमने पढ़ा, फिर वहाँ पे AP endonuclease, exonuclease आता है, and then they remove the entire A basic base pair over there, right? तो वो सब में कैसा होता है ना, confusion आ जाता है. Now we are going to discuss about the nucleotide excision repair. इसमें और हम 10 प्रोटीन पढ़ेंगे. So it's better you make a chart of it, or get the printout of it, ऐसे अपने दीवार में लगा लो, whatever is comfortable with you, just put it over there so that whenever you see, at least remember, okay, mismatch मतलब हमें mute S and mute L, mute H के बारे में है, base में glycosylase के बारे में है, ठीक है? Just an advice, or whatever is comfortable with you guys, you can do in that way. Okay. So unlike base excision repair, the nucleotide excision repair enzymes do not recognize any particular lesion. So if you remember, in the base excision, we have seen that if there is a distortion in the newly replicated DNA strands, this is recognized by the enzyme, right? So rather, the system works by recognizing distortion to the shape of the double helix. यानी कि क्या? पूरा का पूरा shape of your double helix DNA is getting distorted, and that is what is being recognized. Okay. Now, such as those caused by the thymidine dimer or by the presence of a bulky chemical added on a base. Okay. So now you can see from here, you can understand that if suppose there was a ETBR intercalating dyes or any alkylating agent was present, then these are getting repaired by the mechanism of nucleotide excision repair. ठीक है? अभी समझ में आया? अभी पता चल रहा है कि किस तरह का डैमेज कौन सा वाला रिपेयर मैकेनिज्म करेगा, ऐसा आपको कोरिलेट करना है. ओके. Now such distortion trigger a chain of events that lead to the removal of a short single strand segment that includes the lesion. So what they're saying is that जहां पे भी गड़बड़ होगी, that particular patch will be removed and replaced. So this removal creates a single strand gap in the DNA filled by the DNA polymerase, and later ligase will come and it will restore the original nucleotide sequence. Okay. So what are the proteins which are involved in this? So those are known as UVR A, B, C, and D. Okay. Now, if you see that UVR, we are saying. So what do you think, guys? किससे related है ये थोड़ा? Can you guess to which it is getting related? Okay, we'll come to that. आपको फिर idea आ जाएगा. Okay. So nucleotide excision repair means we are talking here about the UVR proteins. Okay. So a complex of 2 UVRA and 2 UVRB molecules, it is going to scan the DNA. Now, in case of mismatch, which protein was scanning the DNA? Mismatch repair में कौन सा protein scan करता है? तन्वी? Akshaya?
Mutes.
Yes, it is the mutes. Okay. So similarly, UVRA and UVRB molecules are scanning the DNA, with the 2 UVRA subunits responsible for detecting the distortion to the helix. Okay, this is important point. This you need to remember. Now, once there is an encounter with the distortion, UVRA exits the complex, and remaining UVRB melts the DNA to create a single-stranded bubble around the lesion. So this much is clear. Together UVR A and B is scanning the DNA, जिसमें से UVR A जो है वो distortion को detect करेगा. Once it is being detected, UVR A leaves the DNA. Okay, then UVR B is going to melt the DNA and making into a single stranded bubble around the lesion. Next, it is going to recruit UVR C. Now, UVR C is going to create two incisions, one which is located 4 to 5 nucleotide 3' और another one 8 nucleotide to the 5' region. ठीक है, अगर जैसे कि यहाँ पे कुछ गड़बड़ है, okay, so this is in 5' and 3' direction. So यहाँ से it's like 3' it is going to take 4 to 5 nucleotides distance और यहाँ से 8 nucleotide distance. So it is making a cut over here. ठीक है, जहाँ पे भी उसने recognize किया who UVRC. Is this much clear, Akshaya?
Yes, ma'am.
Okay. Please remember, huh? Then we'll do along with the figure also. Now this cleavage creates around 12 to 13 residue long DNA strand that contains the lesion. This lesion containing strand is removed and from the rest of the DNA by the helicase, then UVRD. Okay, यहाँ पे कौन सा helicase आ रहा है? It is not the helicase which is responsible in the replication. यहाँ पे आपका UVRD is acting as a helicase. This is resulting in the 12 to 13 nucleotide long gap. Finally, Pol1 and ligase will come and fill the gap. Okay. Now let's see with the help of the diagram. It is the same thing. So what do we see? That there is a distortion first. Yes, we saw the distortion is there. Correct. Now UVR A. 2 subunits of A and 2 subunits of B is coming. A is responsible for detection. It is responsible for the detection of the distortion. Okay, जहाँ पे भी mismatch है. Now, after this detection, UVR A is going to leave the DNA helix. Okay. Now B is going to melt the DNA. It is going to melt the DNA. Okay. Later, it is going to recruit UVRC. And what was the function of UVRC? To make a nick, right? 2 incision. It is going to make 2 incision. एक 5' से 8 nucleotides और 3' से around 4 to 5 nucleotides. So together around 12 to 13 nucleotide long एक gap बनाएगा. ठीक है. Now later what is happening? DNA helicase UVRD is coming and making it into a single stranded bubble. Correct? Now what is happening? The strand is being removed. देखो जिसमें जो गड़बड़ थी, that particular single stranded DNA has been removed. Later your DNA polymerase 1 is coming, synthesizing, adding new nucleotides. Later ligase is coming and resealing the gap. Okay, so this is how your nucleotide excision repair is being done. Is this much clear? Yes, both of you, Tanvi and Akshay, it is clear?
Yes, ma'am.
Yes.
Okay, good. It is simple, right? Okay, so now, now this is the same thing in eukaryotes. It is the proteins are XPC and XPA, XPD. Okay. Now there's something why it is XP, because in case of eukaryotes, especially mammals, xeroderma pigmentosa. कभी सुना है? ऐसा लगता हुआ सुना है कभी? Xeroderma pigmentosa.
Yes, ma'am.
हाँ, so what kind of problem is this in the eukaryotes?
Ma'am, is it vitamin A deficiency?
कौन सा?
Vitamin A का deficiency?
Vitamin A deficiency.
No, ma'am. Ferric.
सोचो. See, already said xeroderma. Derma. किससे related होना चाहिए?
Skin, ma'am, skin.
Yes, skin. So, अभी कुछ आ रहा है idea? So, when you say derma, dermatologist, it is related to skin, right? So, xeroderma pigmentosa or pigmentosum, okay, it is actually a skin disorder. It's a skin disorder. ठीक है, हमने derma बोला ना, so it has to be skin disorder. Now, in this case, what happens is that they are the humans who have got this XP का disorder. They are highly sensitive to sunlight. इसका मतलब क्या? The moment they go out, they get burns in their skin. Okay, they are so sensitive to the minute fraction of UV light which is present in the DNA. Sorry, UV light which is present in the sunlight. They are very sensitive. तो ऐसे केसेस में क्या होता है, जो लोग होते हैं, दे व्हेन दे गो आउट इन द सनलाइट देयर स्किन स्टार्ट्स बर्निंग एंड दिस ब्लैक पैचेस आ जाते हैं, एंड दीज पीपल आर वेरी वेरी मच prone to skin cancer, to develop skin cancers. Okay, so these people cannot go in the sunlight. Okay, clear?
Yes,
Clear?
ma'am.
Okay. Okay, so let's see. So the principle of nucleotide excision repair in higher cells, so you guys like textbooks में NER करके mention होगा. Okay, so don't get confused, it is the same thing. Nucleotide excision repair in higher cells is much same as that in the E. coli. The machinery of detection and exercising the repair is much more complicated. क्यों? See, it's always like that. जितना ज़्यादा वो higher system में जाओगे उतना ज़्यादा complications आएंगे. And complication means it is going to include more number of proteins involved. Okay. So among these is XPC, which is responsible for detecting distortion. अब देखो, UVR A in E. coli is similar to XPC. Okay, it is not XPA, it is C. As in E. coli, DNA is open to create a bubble around the lesion. Formation of bubble includes the activities of helicase, that is A and D, which is equivalent to B over here, and single strand binding protein RPA. So if you see the order, जो हमने E. coli में A, UVR, A, B, C, D पढ़ा है, यहाँ पे वो सिस्टम नहीं रहेगा. यहाँ पे हमारे पास सबसे पहले क्या आएगा? So we are going to have A की जगह होगा XPC, B की जगह है हमारे पास XPA and D. Okay. Now why? Can you tell me why it is like this? कि यहाँ तो हमने ABCD alphabetically पढ़ लिया, but in the case of eukaryotes it is random. ऐसा क्यों? Any idea? This is not 50, it is 5' and this is 3'. So the bubble creates a cleavage site is 5' to the lesion for a nuclease. एक 5' में आपका ये nucleus है XPF and 3' में this is a nucleus XPG. Okay. यहाँ पे वहां पे था आपका 12 to 13 nucleotide long. Here it is 24 to 32 nucleotides long. Clear? Okay, so now the question was why it is like randomly in the case of the eukaryotes. Actually, it is not random. It's that when scientists are working, okay, when the researchers are working, जो protein पहले purify होके characterize हो जाता है, they get the alphabet A, B, C, and D. ठीक है? So there are, in the case of eukaryotes कैसे होता है? First they might have purified the another one and उसका नाम हमने XPA रख दिया. But later we came to know that XPA is actually similar to C action, or we got XPG, which is having activities related to something else. So that's why the order is different. Are you getting the point?
Yes, ma'am.
हाँ, जिसने जैसा जो क्या कहते हैं सबसे पहले प्यूरिफाई करके कैरेक्टराइज कर दिया, हम उसको A दे देते हैं. फिर सब्सिक्वेंट बाद में हम B, C, D देते रहते हैं. लेटर वी सी ओके पहले तो आपका ये प्रोटीन आता है तो पहले G आता है, फिर कभी F आता है, कभी E आता है. ओके, so the occurring of these proteins in a process or in a pathway is different as compared to the procedure or the purification which was done by the other people. अभी suppose you are in a lab, ठीक है, हम सब काम कर रहे हैं, we all three are working as a colleague in the same lab. Now whoever has got the more experience will be able to get the result fast. उसको टेक्निक आ जाएगा जल्दी करने का, है ना. तो अगर जैसे हम तीनों को एक ही असाइनमेंट मिला है कि एक ही मैमेलियन सिस्टम से आपको डिफरेंट प्रोटीन्स प्यूरिफाई करने हैं, तो मे बी तन्वी इज एबल टू प्यूरिफाई वन प्रोटीन अर्लियर, ठीक है, एंड देन शी इज प्यूरिफाइड, कैरेक्टराइज, हमें स्ट्रक्चर मिल गया, हमें सब कुछ मिल गया. सो शी हैज गिवन दैट प्रोटीन का नंबर, लेट अस से दैट तन्वी ने T1 दे दिया, उसको अपने नाम के हिसाब से उसको T1 दे दिया. ओके, लेटर आई गॉट सम प्रोटीन, सो मैं उसको क्या कर दूंगी, मैं उसको R1 दे दूंगी. तो सिमिलरली तन्वी विल स्टिल कीप ऑन वर्किंग, तो वो T1, T2, T3 देती जाएगी, मैं R1, R2, R3 देती जाऊंगी, अक्षया A1, A2, A3 देती जाएगी. लेटर व्हेन वी सी द पाथ कि कौन सा प्रोटीन कब आता है, देन वी सी ओके पहले A1 आ रहा था, फिर R2 आ रहा है, फिर R3 आ रहा है, फिर T1 आ रहा है. तो ऐसे हो जाता है. एंड देन student like us, we have to remember the path in that way then. ठीक है, so this is like already being done. So हमें अभी फिर उसी pathway को हमें यूनिवर्सली it has been accepted, okay, everywhere this has been accepted. उनको नाम दे दिया गया all over the world कि इस प्रोटीन को इसी नाम से जाना जाएगा. So that's why then we have to refer them in that order only. Okay. Now, as the names imply, UVR, they meant to damage the they needed to mend the damage which is from the ultraviolet light. So when I said UVR, it is related to the ultraviolet radiation. So उसको repair करने वाला आपका proteins हो गया है. So when I said that it is sensitive to sunlight, है ना, वहीं से आपको hint मिल जाना चाहिए था. Okay, so they are going to remove thymine dimer or thymine cytosine addicts. Now in future अगर आपसे कोई पूछता है या एग्जाम में आता है that in the eukaryotes or in the humans you get a xeroderma pigmentosum problem, okay, so you should understand that it is related to the sensitivity, hypersensitivity to the sunlight because of the ultraviolet radiation. Because the moment sunlight falls on them, they start, their body or the cells, the DNA starts forming the thymine or thymine cytosine addicts, which is not being repaired in the cell, and that's why they are having this xeroderma pigmentosum ka problem, disorder. Okay, we people are able to repair if suppose it all is happening, so we are not getting those skin disorder or skin cancer. Is this much clear?
Yes, ma'am.
Huh?
Yes,
Okay.
ma'am.
Now, okay, so not only nucleotide excision repair capable of mending damage throughout the genome, but it is also capable of rescuing RNA polymerase, the progression of which has been arrested by the presence of a lesion in the transcribed strand of a gene. Now, what here they are saying is, apart from NER, apart from repairing the damage which is caused by the UV, ultraviolet radiation, it is also escaping the RNA polymerase. What does it mean? See, कभी-कभी क्या होता है? Because of the problem in the अभी transcription में क्या होता है? You have got the bubble again, and यहाँ पे आपका RNA polymerase आके synthesize करता है mRNA. ठीक है? RNA polymerase is making mRNA using this as the template strand. Correct? Now, sometimes because of the problems, like in the something is wrong with the strand, or what happens is that RNA polymerase gets stuck over there. वो वहां पे hold हो जाता है, ठीक है? So now वो आगे जा रहा है. Now it is leaving the strand. कि you can have a truncated mRNA and all. So वहां पे वो hold हो जाता है. In order to remove or escape the RNA polymerase from this stuck transcription, your NER is playing an important role. Okay. It is involved in the recruitment to the stalled RNA polymerase of NER repair proteins. Okay, let's see that. So the significance of transcription-coupled repair is that it focuses on the repair enzymes on DNA being actively transcribed. So RNA polymerase serves as another damage-sensing protein in the cell. So we'll see how. Okay, so this is called transcription-coupled DNA repair. Okay. So what is happening? Can you see? यहाँ पे आपका template, sorry, not this one. 1 second. Okay. This one is used as your template. Okay. And you can see that mRNA is being synthesized. Can you see? Itna clear
Yes,
hai?
ma'am.
Later, they see that there is a distortion. अब आपका क्या होगा? Transcription bubble यहाँ आके stall हो जाएगा. यहाँ पे stuck हो जाता है. So your RNA polymerase आगे भी नहीं जा रहा है, mRNA भी नहीं हट रहा है, so it's getting stalled over there. Then your again NER proteins are coming and escaping this problem. Okay. So upon encountering the lesion in the DNA, RNA polymerase stalls and the transcription stops. So this RNA polymerase is going to recruit your nucleotide excision repair proteins at this site where this distortion is being seen. Okay. So then these proteins will come, remove the RNA polymerase, and they will start their repair mechanism. So even the transcription is helping in detecting the distortion or any mismatch in the DNA, and it is helping in repairing with the help of NER. Okay. So far so good. सब समझ में आया है?
Ma'am.
हाँ.
So if it stops abruptly like this, so will it start again from that point? The transcription.
Yes. See, if the gene is required, okay, if the gene expression is required, now if this problem has occurred, it is going to get it repaired, and then again it will go back and find its all promoter jahan se shuru hua tha, and again it will start. Okay. Yeah. Now, since you have asked me this thing, one interview question I remember now. So I was just sitting in the panel, and next to me, the scientist, he was asking this question to one of the girls, okay, who came for the interview. So he said that you are going to do transcription in a test tube. Okay. So transcription is need to be carried out in test tube. Now, the question was, what all are the requirements in order to start the transcription? So guys, what do you think? What all are the requirements? So I know I haven't taken transcription yet, but there will be a few rough idea, right? सबको मालूम होगा ट्रांसक्रिप्शन करने के लिए क्या क्या चाहिए होता है. तो अगर मुझे ट्रांसक्रिप्शन एक टेस्ट ट्यूब के अंदर करना है, ठीक है, विदाउट सेल, so it is also called cell free system. आप प्रोटीन ट्रांसलेशन भी कर सकते हो और ट्रांसक्रिप्शन भी कर सकते हो. ओके. तो अगर मुझे ये टेस्ट ट्यूब में करना है, मैं इसमें ऐसा क्या क्या ऐड करूं जिससे it will carry out the transcription. Of course, you need.
Yeah, it means DNA.
You need DNA as your template. Okay. Then what else?
We need tRNA.
This is transcription, not translation.
Oh, sorry. Yes.
One negative mark. Then और क्या लगेगा? Of course, RNA polymerase. Then mRNA बनाना है तो और क्या लगेगा?
Amino acids.
अरे बाबा, this is transcription. Translation में amino acid लगता है. So here you are going to need RNTPs.
Oh, yes.
Okay. mRNA, ribose nucleotides, then you need amino acid translation में लगेगा. Then again -1. और क्या लगेगा? तन्वी? Then what is the starting point for any transcription to begin? So DNA का gene हो गया. So what all should be present on this gene so that the RNA polymerase can come and start?
The promoter region.
Yes, very good. So that is a word which we wanted to hear. Okay. So promoter लगेगा, किसी में operator रहेगा. Okay. So all those protein requirements which will go and bind to the promoter operator region, so that the RNA polymerase or the transcription initiation complex जो बनेगा, वो सब जो आपको requirement है, that you guys have to tell. ठीक है? So RNTPs is of course required, RNA polymerase is required, but in order to start the transcription, what all is main important things are there? So promoter, operator, ये सब हमें लगेगा. अभी see inside the cell, they can form a hairpin structure, loop structure, and the promoter operator can be bring together. Okay, but if you are doing in the lab, in the test tube, then all these things should be present in the gene. Then only those required proteins, RNA polymerase can come and they will bind and they will start the they will start the transcription. Okay, good. So main important point was that you need to talk about the promoter region. Okay, good. But don't get confused with the transcription and translation. Okay, so don't just say amino acid chahiye ya tRNA chahiye, सब कुछ नहीं चाहिए. Okay, so central to the transcription coupled repair in eukaryotes is the general transcription factor TF2H. So अभी के लिए थोड़ा सा ऐसे याद रख लो that we are just talking about the TF2H, that is transcription factor 2H. We will be talking about this in transcription as well. So TF2H, it unwinds the DNA template during the initiation of transcription. So even this is what you have to talk when we say that you have to do the transcription in the cell test tube. ठीक है? So हमें एक helicase बोलते हो, unwinding protein भी चाहिए, right? Okay. So the subunits of TF2H include DNA helix opening proteins called XPA and XPD. Remember what was the function of XP and XPD? They are related to the function of UVRB, which melts the DNA. Yaad aaya? Okay. So along with this, your TF2H is also working. Now it is responsible for two separate functions. First, it stands separating helicase activity, which melts the DNA around the lesion during NER. Okay, and also it helps in opening the DNA template during the process of gene transcription. याद रहेगा? बहुत सारे proteins आज हमने discuss कर लिए. Are you able to recollect? Yes, guys, able to remember? थोड़ा बहुत तो याद रह रहा होगा. Okay, let us just summarize. Okay, तो हमने सबसे पहले क्या पढ़ा? Let's talk about E. coli or prokaryotes. So सबसे पहले we have talked about, yes, Tanvi, would you like to start?
Yes, ma'am. Ma'am, about UVR, that?
हाँ. Yes. So we started with NER today, right? And in which we discuss what all proteins we have discussed today.
Ma'am, UVRA and UVRB and UVRC, then
D.
that makes
Do
the
you
nick.
remember? Do you remember the function of A, B, C, D?
Ma'am, UVRA and UVRB detect that distortion.
A does
And
the
ma'am,
detection of the distortion.
yes, ma'am, and UVRC makes the nick. UVRD, ma'am, maybe some helicase.
Which separates the single stranded DNA from the rest of the DNA. B क्या करता है? Melt करता है. Okay.
Melt.
Good. So this was in the case of the E. coli. Similarly, we have XPC, XPA, D. Okay. So C का काम क्या है?
Ma'am, same as UVRA.
And A and D are doing something which is similar to melting. Separating the single stranded DNA. If you remember in the figure, we have shown that ऐसा कुछ रहेगा. And जहाँ पे भी lesion था, that particular strand is getting removed totally. And यहाँ पे नया 3-OH आएगा and polymerase 1 will come and add the new nucleotides, and then ligase will come, which will really seal the gap. Okay. So in case of eukaryotes, apart from XP, A, C, D, B, we have TF2H also. That is transcription factor 2H. ठीक है? ये additional आपको याद रखना पड़ेगा. Okay. So it is having two functions. A, which is around the lesion. Okay, it is helping in separating the DNA around the lesion during the NER. So जो हमने पढ़ा. Okay, NER में जो strand separation करता है with the help of your XPA and XPD, even along with XPA and D, TF2H is also helping. Plus, it is also required during gene transcription. So gene transcription जो हम करेंगे in further lectures, we will talk about TF2H. Right now, what you have to remember is that along with your NER proteins in eukaryotes, we have discussed XPA, C, D. Along with this, you also have transcription factor 2H. Itna clear hai?
Yes.
Okay.
Yes.
Okay. And we have also discussed that NER is also helping in the transcription, which is getting stalled because of the distortion. Okay. So जहाँ पे भी RNA polymerase is getting stuck because there is a distortion in the DNA, then also it is going to recruit your NER so that it can repair the distortion. So in a way, it is like, after the replication, if suppose there are still a mismatch which was, escape from these repair mechanism, So during the transcription, if there is a distortion detected by the RNA polymerase, then it is going to recruit the NER protein so that it can repair. Okay, so this is known as transcription coupled NER. Clear?
Yes, ma'am.
Okay, yeah. So just join me again.